Whoa, hello. Okay, good morning, everyone. And today we're going to talk about how DNA is our genetic material. Now, I know we know this, but there used to be a time, not so very long ago, where they weren't so sure about this. So, by the late 1800s, scientists had a limited knowledge of genes. So they knew that heritable information is carried in these little units called genes, and they knew that genes determine inherited traits. Okay. But they didn't know sort of what a gene was. So in the early 1900s, scientists figured out that genes are parts of structures called chromosomes. Okay. And they knew that chromosomes were made of DNA. Right. So how they did this was through a couple of key experiments, and you have to know these experiments. So Griffith did the first experiment. Griffith worked with two strains of Streptococcus pneumonia bacteria, so pneumonia. So there were two strains. The S strain caused pneumonia when injected into mice, and that killed them. The R strain, okay, did not cause pneumonia when it was injected. The mice were just fine. Okay. And so S strain causes pneumonia, R strain doesn't cause pneumonia. Then if he took the S strain and killed it with heat, he denatured all its proteins, and injected into mice, it did not cause pneumonia. So yes with this, no with this, no with this. There's going to be a sum up slide in a second. So then Griffith did something that kind of really was a tipping point. So he took the heat-killed S strain that does not cause pneumonia, and he mixed it together with the R strain that does not cause pneumonia. And when he mixed the two of those together and injected them into mice, they got pneumonia and died. Okay, so this was, I got to say, unexpected. I wish I had been there and they were like, what is happening? So neither strain caused pneumonia on their own, but together they caused pneumonia. So here's a little summary. So R strain, you inject it into mice, the mice are totally fine. Okay. S strain, okay, you inject it into mice and it's, not so good. If you heat kill the S strain, denature all the proteins, and inject it into mice, they are happy, fine, does not cause pneumonia. If you mix together the two, so this one and this one, that don't cause pneumonia, when you inject it into the mouse, they die again. Well, it's new mice. So the moral of the story is, and this is the key box down here, something from the heat-killed S strain can transform the harmless R strain into a deadly S strain. Okay? And this was kind of the link. Okay? So transformed bacteria, and we'll talk about what transformation exactly is later. But you know what a transformation is, like a transformer. It starts off with one thing and changes into a different thing. So the results of Griffiths experiments led to a couple of new deductions about genetic material. So some substance in the heat-killed S strain changed the R strain from being harmless into being deadly. Okay, that Griffith called this, he coined the phrase transformation. Okay, so, and the substance that caused this transformation, they thought, is the long sought after molecule of heredity. And they were right. And it turned out it was DNA. Okay, but they weren't quite sure of that yet. So, excuse me, so sorry. Another team of scientists took the work that Griffith did and built on it, and they were these three guys, two guys and a woman, so Avery, McCarty, and McLeod. McCarty is female. They isolated DNA from the S strain bacteria and mixed it with the live R strain bacteria and produced the live S strain bacteria. Okay, so this suggested that the transforming molecule was DNA. So to rule out the possibility that there was protein in there contaminated it, Avery treated the samples with enzymes that destroyed protein, and he still got transformation. Okay, when he added enzymes that destroyed DNA, there was no transformation. Okay, and that kind of solidified the fact that our transforming molecule is DNA. So, and that can, of course, explain Griffiths experiments. So if you heat S strain bacteria, it kills them, but it mostly kills the protein. It doesn't destroy the DNA. So when you take those killed S strain bacteria and mix them with the living R strain bacteria, fragments of DNA from the dead S strain become incorporated into the chromosomes of the R strain. Okay? If these fragments contain genes that need to cause disease, the R strain will be able to cause disease. Okay? So between Griffith and Avery, McCarty, and McLeod, they concluded that genes are made of DNA. And this is just kind of a diagram that shows you how transformation happens. So here we have a bacteria. Here we have some DNA fragments, okay? And they get bloop, sucked up into the bacteria. Here they are. Then they get incorporated into the DNA. And then they are part of the DNA. And they can change or transform the nature of the bacteria. So it changed it from R into S. Okay? That's all for how we basically figured out that DNA was the genetic material.